safety in synthetic biology are coming up now a lot more.
Hawks, I am a professor of anthropology at the University of Wisconsin-Madison.
And I'm Christina Agapakis, and I am a graduate student at the Harvard Medical School.
Wow. Okay. Christina, the exciting news of the past couple weeks, I guess, in biology has been related to Craig
chromosome. And there's a lot of people working on making sort of biological circuits in yeast also.
As far as anybody goes, for that matter. So can you tell us what exactly the announcement entails?
No, basically the announcement is that the J. Craig Venter Institute has synthesized a whole genome of a bacteria and used that chromosome. And there's a lot of people working on making sort of biological circuits. And so,
Okay. So the genome that they're using, they made it up?
No, they used a naturally occurring sequence and just sort of remade the natural sequence using chemicals synthetically. But the sequence is almost exactly the same as the naturally occurring one. They did make a few changes here and there. I think they deleted 2 genes, and they added a watermark that was sort of a little poetic interlude and a little message sort of saying the JCVI was here.
Wow. Okay, so for those of us who sort of know a little bit about that recombinant DNA technology, what's different between this and what you would do if you were cloning a bacterial cell?
I think the main difference is really scale here. So people have made synthetic genes before and put them into bacteria and had them function in bacteria, but in this case, it's the whole genome, not just a single gene or bacteria. But in this case, that's really the main difference. Here, but in this case, it's
okay.
Yeah,
I heard that some of the stuff that they put in was like James Joyce verse.
Yeah. I thought that was actually really sweet and charming. I don't remember the quote exactly, but it's something about from life to life and really sweet and charming. Being human or something. It's actually really sweet and charming.
Okay, so what's the next step? I mean, this is they've got a colony of cells going that has their DNA in it that they designed. What's the next step? What happens next to these cells?
Well, these cells just hang out, I guess. I think I'm interested in sort of seeing what kind of new genomes we can make and how we can sort of combine genomes from different organisms to make something new and different and maybe more useful than this sort of very slow growing and small bacterium. I think there's a lot of other things that we can do that are really interesting with the technology. Something that we've sort of been talking about would be an interesting next step would be to synthesize a genome that's something that we've encoded in a different code. So basically, something that we can make it so that the amino acids that are coded for are sort of matched. So there's a genetic code that's shared between almost all organisms that have 3 bases of DNA make 1 amino acid, and so you could make a different code basically by if you can synthesize it yourself in a computer and then put it into a bacteria, that bacteria could now start making a genetic code that is unreadable in a computer by other organisms.
Okay, so let me get this straight. I mean, the basic process of translation of DNA sequences into proteins is you make a messenger RNA, and it's got a complement of the DNA sequence, and it goes out and collects, when it goes to the ribosome, transfer RNAs are carrying around amino acids, and they connect to that in order according to these 3 base pair codes. And so what you're saying is that you'd redesign the transfer RNAs so that they recognize a different code but carried the same amino acid? Is that...
Yeah, that's the idea. So people have done it before, sort of on a small scale. You can design tRNAs to carry and encode sort of non-natural amino acids. But you can sort of now imagine at a larger scale, if you can synthesize the whole genome, then you could make a whole genome that is now sort of unreadable by other organisms, and in that way, actually, would be safer. A lot of the concerns about safety in synthetic biology are coming up now a lot more. Genome that's unreadable by other organisms, the DNA would be something that would sort of be essentially harmless because it can't it can't be read by anything else. So if there was a transfer of DNA, it wouldn't, nothing would happen.
Okay, so for the evolutionary biologists out there, they would probably think, well, there might be some optimization that's happened in the course of evolution, that this code that we have works pretty well. And so if you design an alternative code, maybe there's some stumbling block. Is there anything that that looks like a barrier to doing this?
Oh, I mean, this is sort of definitely still pie in the sky. We're sort of imagining pie in the sky, where it could go. But I think pie in the sky, sort of the evolutionary argument is interesting, but I don't know if sort of right off the bat there's a reason why. I think it has to be one code or another, and if everything was matched within the same code, then
Okay. Well, let's. I guess it's a natural thing to go on to talk about sort of what your work involves and how to. I mean, synthetic biology is a big area right now. And a lot of people who in the past might have called themselves something else, bioengineers or basic geneticists might today call themselves synthetic biologists. So how do you see that field? I mean, what are its contours like?
I'm sort of very welcoming synthetic biologist. I would say anyone who wants to call themselves a synthetic biologist should go ahead and do that, and will sort of almost retroactively call a lot of things synthetic biology. I think that there's a lot of things that sort of fit in under the idea of engineering, using engineering in biology and using biology for engineering. And so that's sort of where I think that everything that sort of has an engineering slant at all would be synthetic biology. My work personally is sort of about a little bit wackier, sort of thinking of new things to bring into synthetic biology. So wackier, sort of new thinking of new things to bring into synthetic biology. So I'm working a lot on enzymes that transfer electrons in metabolism. Sort of new things to bring into synthetic biology. Enzymes sort of as a tool in synthetic biology, as components of biological circuits. So that's one thing that I work on a lot. So I'm interested sort of much ideas on a much smaller scale. I don't really think about genomes. I think about sort of enzymes and pathways a lot more.
Right. I thought of synthetic biology before. I wasn't necessarily designers. I was thinking
I'm interested sort of much on a much smaller scale. I think that biology.
I thought of synthetic biology before. I wasn't necessarily
thinking about now is sort of building tools that can be useful. So there's people who are working on making medicines and fuels and bacteria, medicines and sort of as a way to make a lot of things very easily and sort of safer and more environmentally friendly than a lot of the chemicals we're working on making as medicines and fuels and bacteria. Oil is a problem. People who are working on making medicines. So I think that sort of idea, there's sort of make a lot of chemicals already. So the idea is just to sort of tweak it, make things a little bit better, a little bit more useful. Bring things into different contexts. So that's where I think making the medicines and fuels and synthetic biology is now sort of as a way to much bigger or smaller or just different things going on too.
Right. I mean, so your basic recombinant DNA technology, from the 1980s is you take a gene from something that already works and you put it into a bacterium and it makes the product.
Yeah.
And synthetic biology is in some sense different because you get to design the genes.
Yeah, so most of the designing of the genes is also sort of changing, making small changes to things that exist naturally. The other, I think the main sort of conceptual difference with synthetic biology as it's sometimes defined versus recombinant DNA technology is that the genes you make sort of a pathway of genes rather than sort of take, you don't take just the gene that makes insulin and put it into bacteria. Now you have the insulin making bacteria. It's more like you have to sort of understand, bring enzymes from a lot of different organisms together, sort of understand how these parts all work together in a cell and sort of in different contexts and sort of move them around and create a new pathway, sort of from components that already exist in nature, and sometimes entirely new components, but that's still very, very hard.
Sure, yeah. Well, when I think you see some press about what synthetic biology does, or what people who are, really active in the field think, okay, where is this going? Sometimes you see them talk about, components. They talk about them almost like Legos.
Yeah.
Like you could make little molecular components that will be parts of machines, and you can reuse them and mass produce them and put them together. Is that something that is realistic, or is it sort of, you mentioned pie in the sky, but is that, where people are going or not?
I think that a lot of these, they're really just simplifications, really, and sort of abstractions as a way to be able to really deal with the complexity in biology. Like, sort of the goal is so that you would be able to take sort of a very well-characterized biological component, sort of an enzyme or a genetic sequence, and say, I know exactly what this is going to do, based on my computer model of the cell metabolism. Now I'm going to go and put it in next to these other components, and this is going to happen, and I know for sure. So that's the sort of one dream of synthetic biology. Right now it's sort of a lot more ad hoc and sort of trial and error. So you take a bunch of components that from different organisms, put them together and say, does it work the way I think it is? Why doesn't it? And seeing how it doesn't work sort of tells us a lot about what we know about biology, just sort of as a science, and can tell us a lot more about how cells work and how these genes and how these enzymes and proteins all work together in a cell to sort of make the amazing things that cells can do.
Well, I think you're definitely right about that. I mean, in the sense that, one of the big areas in understanding biology today, from a genes to phenotype type of level, is how modular are things, and how much do they depend on each other during development. The idea is that you could have a system of genes. No gene works in isolation. They all sort of have to work together. Mm-hmm. But you could have a global system, where every gene potentially interacted with every other gene. Or you could have a very modular system. Sort of like the engine of your car, where the stuff that's in the carburetor doesn't have to interact directly with the stuff that's in the air filter and so on. And, of course, cells are somewhere in between, because there's this big wet area where things are floating around and they have a chance to interact, even if they aren't really designed to interact well together. Mm-hmm. And cells have to tolerate that.
Really sweet and charming.
But if you're designing a system, a chemical system from scratch, you might very well want to design it in a way that was more modular because you could control the effects of it. But if there are barriers to that, if by the very nature of the system you're designing, some kinds of interactions go on that you can't really control very well, then you end up with with the same problem that biology does. How do you make these things optimized?
I think that's a really good point. I think because biology is built out of sort of these shared components, so everything has DNA. The DNA always codes for proteins that are enzymes that interact with chemicals and can sort of interact with each other and make these sort of really complex webs of very, very complicated behaviors. So there are modules, right? So like there are genes that are things that are sort of components.
Oh yeah,
right?
Some are highly modularized. There are some genes that only interact with a couple other genes, and that's what they do.
Yeah.
And then there are some that you would think that's what they do, but then you discover that they have some weird effects elsewhere in the body.
I don't know. It's actually really charming.
This is one of the things in my research that we're always running into. We've got a gene. We know from Mendelian disorders that it does, it's active in some tissue because it causes a disorder if there's a mutation to it. Or we know that if you tweak it in some way, it has an effect. But it could be that tweak it in a different way and it has an effect somewhere completely unpredictable. Hopefully that doesn't happen every time we look at something, but it happens often enough that it makes us sort of pause.
Yeah. I don't know. I think it's so interesting, though, right? Because I think so. You can take this gene that's a short sequence, and then you can move it around, and then you can see all these things that happen and how it interacts with everything else. I don't know. So sort of like as a object, you can move it around of research. It's really interesting. Move it around. One thing can have so many effects, and that's why that's why science is interesting. That's why that's why we're doing it around. That's why we want to study biology. So I don't figure out how this is all working. I don't know. So maybe it'll never be that one part will make one thing. I don't know, but I think that maybe we can sort of, using this kind of understanding and the things that we can figure out from trying all these combinations, I think you can make things that are really useful and really interesting and sort of chromosome. And there's a lot of people working on technology, making sort of biological circuits in this also, so making things like that.
Okay, so on that subject...
And there's a lot of people working on
move it around.
Making sort of biological circuits in
yeast
yeast also.
Can move it around.
Move it around.
And there's a lot of people working on making sort of biological circuits
so there are
in
people
yeast
making
also. So
biofuels in algae and in yeast and in E. coli. And it's just, they're not making the whole genome. And I don't think that you have to make the whole genome synthetically in order to make a successful biofuel. Make the whole genome synthetically in order. I think it's just we're going to do it. This is an interesting sort of as a technology. It's very interesting. We're going to do it very soon. The concept is very interesting, but I don't know directly how useful. I don't know even within the next 10 years how useful designing a whole genome is going to be for biofuels in particular. So I think like their biofuels project is much more sort of traditional synthetic biology in that. Sort of you bring together genes from different organisms and trying to create a new metabolic pathway.
Well, on that level, it's not in principle different from putting a vitamin enzyme into rice or putting insulin into bacteria. So let me ask, bacterial cells. Or prokaryotes generally are sort of simple,
and there's a lot of people working on
but in particular,
making sort of biological circuits in yeast also.
Is there eukaryotic synthetic biology on the horizon, or is this an order of magnitude more difficult?
There are people working on eukaryotes in my lab. There's. Again, it's a much smaller scale. Like no one is synthesizing a whole human genome. Right?
But for example, yeast. Yeah. We've been putting genes into yeast for a long time. But is it? Can we expect to see somebody assemble a yeast chromosome?
People have assembled yeast chromosomes actually. I don't remember the school. Maybe Penn State. I don't remember. I don't remember. But there's an iGEM project and sort of an undergraduate undergraduate spearheaded project to assemble a whole yeast chromosome, and I'm pretty sure that has happened already. So yeah. Okay. Not the whole genome, but certainly a chromosome, and there's a lot of people working on. Making sort of biological circuits and making things. You can make little switches. So if you put in one sugar or versus another, the cells will grow red or green. So those kinds of little smaller systems are certainly being developed. And in my lab, people are designing memory in yeast. So the idea is that to see whether or not the cell can remember having experienced something. So they if you put in galactose, which is one kind of sugar, and then take that sugar away, the cell will remember that it has experienced that sugar by keeping on a positive feedback loop that makes a color, a yellow fluorescent. A positive feedback loop.
Okay, so when you say memory, this is very much like integrated circuit type of memory, where you flip a switch and it retains its state.
Yeah, that's the idea.
Okay. Well, how about we say a few words about the press coverage of the synthetic biology studies? Because at the moment, we've sort of gone through, what this is, what it means, what's the next step. And when I read people writing about this, especially in newspapers or on the internet, obviously, but sort of general public interest journalism, what I get is a little bit different picture of of what's going on. They have a spin that they want to put on it. So what's your perception of that as somebody who works in the area?
I think for people in synthetic biology, nothing was a surprise. So everybody knew that. I think that the Venter Institute was working on this. Everyone knew that it was right around the corner. That they really and I mean Craig himself was saying it's right around the corner. We're going to do it very soon. Like we're almost there. Just a little bit more. So everyone knew sort of knew the story already. And since they had already synthesized the genome, and that was a paper a couple years ago, and they had already transplanted genome from one organism to another again a couple years ago, it was all the pieces were in place, and so nothing was terribly new for us. And then also nothing was new when we saw the news reports that are saying, "Oh my gosh, we've created life, Frankenstein, this and that." So I think most of the stories start with the sort of very sensationalist, move over Frankenstein or the Economist cover.
Oh, of course, our scientists playing God. I saw that headline several times.
The Economist cover with the Sistine Chapel and Adam is poking the cell to start life. You don't need God anymore. I thought that was a little bit too much, but then most of the articles sort of go into all of the caveats.
It could have been worse. It could have been they pasted Venter's face on the God and had him touching the bacteria. It could have been much worse.
Yeah, and I think that's not the kind of press that the Venter Institute wants, and it's certainly they're trying very hard, and I think doing a very good job of sort of presenting what it is and what it isn't. There was an article that they wrote in the Wall Street Journal yesterday, I think, I don't know when this is going to play, but very recently they. And saying sort of, we didn't create new life, we just made a synthetic genome, like this is really interesting and like a sort of a step towards perhaps making synthetic life as a bigger story later, but right now it's just the genome and it's sort of this technological advance. So I think they've been doing a very good job, and most of the articles sort of, if you do keep reading and read more closely and sort of see the reactions from other scientists and all the quotes in there, most people are sort of, saying like it's great, like it's very interesting, and it's a very very impressive technological feat. But let's tone it down a little, and I think that that the story is there if you look the right place.
Well, it's very much mainstream science.
Yeah.
I mean, in the sense that, like you said, the elements are in place. Mm-hmm. I think people had a reasonable expectation that this could be done with what we know today, present day technology. Uh-huh. And there wasn't, it's not like suddenly they're making, power, or suddenly there's not a massive surprise in it. I mean, in that sense, it's a normal science sort of enterprise. Mm-hmm. They did something that everybody thought would be possible, and they proved that it in fact is possible, mm-hmm. and they showed that what you expect to see happens, and it's a great story about how science works.
Absolutely.
But on the other hand, there's this sensationalist aspect that, there's this lone genius and he's doing everything in his own private institute, and there's this new technology and it's fundamentally different from anybody thought was it's more tension in that story than in most.
Yeah, I think it's also the funniest parts are the how Craig has in the race to the synthetic organism, he has won, and I think it was never a race. That was the sort of the most sort of amusing to people in the field. It's like no one else is working on it. Like everyone knew this was Craig's project and sort of his crazy thing. So the Venter Institute is also working on a lot of other things that are more similar to what everyone else is working on, I guess, is the this was always sort of their crazy thing, and it's not a race. I thought that was sort of very funny, but I think I mean Craig has been in the news for a very long time, sort of as this pioneer, and I think that he's done a very good job. Sort of like promoting science and making science as sort of a big story. Like I think that this is fabulous that there's so much news coverage for something that is so not useful. It's like, okay, we have goat mastitis. Terrific, right? Like so, but to have so many people interested in something, I mean, so sort of mundane as a chemistry project, almost is really, I think, is great. And sort of even for me, like going to graduate school and wanting to become a scientist was from when I was younger and, hearing the news about the Genome Project and how, there's this race between Craig Venter and the public funded project and how it's all this drama, and then they're always talking to Bill Clinton and there's all this stuff going on. So I think it's definitely inspiring to me as a young person interested in science, and I hope that that's sort of happening for people now, sort of hearing these stories and sort of wanting to be biological engineers.
Oh, yeah. Sure. It's there's that aspect to it. I mean, there's really, when you're working in science, in something that is important to the group of people who work in it. And when we start talking about the Neanderthals, it's very much a case like this. You've got something where there's a developed science. We all interact with each other. We all know what the basic questions are. We're working on those questions. We're developing knowledge. But this all happens in a very small really club of people. And to have a big story that comes to the public attention can do wonders to be able to, allow you to show people what you do and why it's interesting, why it's important, and also to give them a common reference frame so that when you say, oh, I'm working on this, they have oh, you mean like that other thing. And even if it's not exactly like that other thing, and even if that other thing is not really super important, it's still, it's really important to have that connection, because without the connection, it's, you get the reaction of, oh, well, it can't be that interesting because I've never heard about it.
Well, so is this a good time to transition into talking about the Neanderthal genome?
Sure, yeah, let's talk about this.
The other big news story?
Yeah. So now it's, 3 weeks ago that the Neanderthal genome draft was announced and published. When I say published, they've been talking. It's very much like the Craig Venter Institute. I mean, people know that the Max Planck Institute for Evolutionary Anthropology has been working on a Neanderthal genome draft, and this has been in the works for several years. So, in that sense, it's not surprising. I mean, the schedule was that they should have their. Now it's about 5 billion base pairs of sequence by early 2010. So that's not surprising. Any big project, it's going to be, there's a timetable. And we've come to that point in the timetable where we have a draft. But the draft is now online. You can download it. You can start working with this stuff. And so there is a real exciting announcement type aspect to this. And that is the availability of the information. And, of course, the preliminary sort of analysis of it, including the big news story announcement, which was many of us descended from Neanderthals. So that's, in anthropology, it doesn't get any bigger than that. I mean, this is big news.
Yeah, so I guess, why is it big news? I saw in the article about the genome that it was... that there has been a debate for many years whether or not there had been any interbreeding between Neanderthals and humans, and I guess I don't know a lot about the sort of the history of that debate. Is that something that's sort of why is it interesting?
Why is it interesting? Besides the sex aspect, right? Yeah, because everybody knows Quest for Fire. There's. The thing is that there are these ancient people who lived in Africa and other ancient people who lived outside of Africa before 30,000 years ago. And the Neanderthals are one of these groups of people. They lived in Europe and in West Asia, really as far as the southern tier of Siberia. And there were other groups of people who lived outside of Africa, and there were probably several groups of similar scale in terms of numbers and geographical extent living inside of Africa. That seems to have been the population structure of our species. And we know something about these people from their archaeology. We know that they were making different types of tools in different places, that there was a certain pace of change, which in today's terms was a very slow pace of change. But over tens of thousands of years, technologies transitioned one from the other. And we know that there was regionality, so that in different parts of Africa during this time period, which in Africa we'd call the Middle Stone Age. There were different traditions of stone tools. There were different traditions in Europe, different traditions in Asia. And so we sort of understood that there are these people. We know that they're simple hunter-gatherers. Their technology at that time was, compared to anything that we have in industrialized societies today, obviously very rudimentary. But it was along the same lines of some living recent groups of hunter-gatherers. So this was human technology. It wasn't like, some sort of transitional half-human, half-ape technology. It was human technology. The question is, how did they interact? Now, gosh, it's been almost 30 years. And really, it's been almost 40 years since the first demonstration. Humans have relatively low genetic variation. If you looked at humans today, obviously there's more than 6.5 billion people in the world. But if you look at humans genetically and say, let's assume that humans are just mating at random and their genes don't do anything interesting, so they're all evolving like under genetic drift, how many people would we say that there were based on how variable we are? And the number that you get when you do that is something like 10,000.
Wow.
So this is a big difference, right? You have to account for this difference somehow. 10,000. Were there 10,000 people at some time in the past? Did we have a founder effect? That there was a small population somewhere, and that population grew and spread, and if so, where did that population live? What kind of geographical extent would it have? And over the course of the past, really 30 years, people have been saying, well, if this is the explanation, there were a really small number of people. They couldn't have lived in all of the places where we know that fossil humans and their archaeological remains are found. They have to have lived in some small place. And the small place, because Africans today are much more genetically variable than other populations, the small place was thought to be Africa. And in today's terms, looking back and saying, okay, now we know that they weren't just living in a small place. We know that we have this genetic contribution from Neanderthals at least, and possibly some of these other groups of people. What do we interpret about this effective population size is what we call it now. What we'd say is that there's got to be some sort of breeding structure that has led to our very low variation. And in addition to that, we do know that only a small fraction of the genes of today, people who live outside of Africa, only a small fraction of their genes come from Neanderthals. It's something like 1 to 4%. So, 1 part out of 30 or less.
And it's different parts too, right? It's not always the same genes. Is that true? I thought...
That's right. These are genes that vary in humans today. They're genes that are not fixed outside of Africa. So that means that you might have a different set of these than somebody else. Your proportion is going to be the same because we're averaging out over 3 billion base pairs of DNA. It's a big enough number that your actual fraction is not going to differ very much. But it could be something different in you from somebody else. We don't know at the moment whether any of the things that we got from Neanderthals are important or functional, or whether they do anything. Some of the obvious candidates, you look at people outside of Africa today and say, how are they different from people in Africa? I mean, if people outside of Africa have more Neanderthal genes than people in Africa, I say more because we don't really know whether there was gene flow coming back into Africa from Neanderthals. What we know is that the people outside of Africa today are more similar to Neanderthals than the people who live in Africa. So that tells us that there has to have been a contribution to that population, but it doesn't tell us that there couldn't have been a contribution to both populations that has averaged out. But so we don't know whether their genes do anything useful. My guess would be that some of them probably do. The obvious ones, things like skin pigmentation. At the moment, there's no evidence that today's skin pigmentation genes that influence lighter skin outside of Europe, there's no evidence that any of them came from the Neanderthals.
Okay.
Maybe we'll discover that there's some that account for some proportion of variation that do. But at the moment, we don't have any evidence that they do. But we're talking about an ancient group of people that was evolving on its own trajectory with less connection to Africans and other populations than today's populations have. They should have had some things that would have been useful, potentially, in later populations. Things that might have nothing to do with where they lived, necessarily. Things that have to do with metabolism, maybe what they ate. Or things that have to do with the diseases that they might have faced. Stuff that wouldn't be obvious in the paleontological perspective. We might discover something interesting about that. So, it's exciting stuff. I mean, this is stuff that my lab is working on. We're trying to find what genes do we have from them, what do they do, is there any pattern, is there anything interesting, and what kind of interactions can we say on the basis of our similarities and differences from them? What kinds of interactions can we exclude? What hypotheses can we test about these interactions?
Yeah. So the one thing that struck me the most from the paper and from sort of reading is just how similar the modern humans are to Neanderthals, and how, I think you said something, how within the variation within the Neanderthal genome, or like group different individuals, is bigger than differences between modern humans and Neanderthals, or like any individual modern human, or something like that. Is that true, or am I getting it wrong? They're so similar.
I'll give you a short version. So if you compare humans and chimpanzees today, we differ by about one base pair out of 50. So we're about 2% different. If you compare humans with other humans today, we differ by about one base pair out of 1,000. So we're between 10 and 20 times more similar to each other compared to what we are with chimpanzees. So if you think of this in terms of years, we think that we divided from chimpanzees, our population split from them something like 5 million years ago. Well, that means that humans, on average, 2 human genes split from each other in ancestry something like 500,000 years ago.
Okay.
If you look at us and Neanderthals, they're different from us by just a shade more than we're different from each other. So that if you compare their genes and our genes, looks like about maybe 800,000 years is the average. However, that's recent enough that there are some human genes for which 2 human copies taken at random are more different from each other than one of them is from the Neanderthal. Blood groups are like that. The ABO blood group in humans looks like the O allele originated something like 2.5 million years ago for the first time, and there are several different O alleles because this is it's a non-working copy of this, and so different mutations have caused this non-working copy over the course of the past. The first one 2.5 million years ago. So some people for ABO, and A and B separated something like 3 million years ago. So people who are A and B, people who are A and O, are more different, those 2 copies from each other, than the O is from a Neanderthal, because the Neanderthals that we have so far are all type O.
That's
and
interesting.
originated, we think, in the same event. So that's a case where you say Neanderthals actually share something with us that's, phenotypically not obvious. You wouldn't look at them and say, oh, well, they're all type O. But genetically, it's something that's highly salient. I mean, everybody's heard of this. If we look at their population and our population, they're a little bit less variable, taken collectively, than we are. So they look like they've got a little bit less variation. Now, the 3 nuclear sequences that we have the most information from are all from the same site. So you could say, well, okay, yeah, they're more genetically similar, but what if you took 3 people from the same city today? That's the proper comparison. There, it's not obvious. Their mitochondrial DNA which of course is a small fraction of your total DNA complement, but it's useful because we know a lot about its variation today. Their mitochondrial DNA is less variable than humans by quite a bit. But if you compare humans that today live in the same geographic range, it's the Neanderthals are only less variable by a little bit.
Okay.
So that's more or less comparable between.
Is it something in your lab? Do you do any sequencing? Like, do you have to use clean rooms to make sure that you don't contaminate the?
Oh no, we don't do any wet lab stuff here. I'm a population geneticist, basically. So my role is to figure out what these gene sequences mean. And from the point of view of from my point of view, having a genome draft is suddenly I got into this by studying fossils, and I know all these fossils, and there are a couple of hundred good Neanderthal specimens where good means that you've got more than a tooth.
How can you tell that this is another question. How can you tell that the bones come from Neanderthals versus other ancient humans? Is it just like from the context within which they're found, or is there something different about the bone, or do you have to do any sort of sequencing or dating, or I don't know, sort of more?
It's like fossil forensics. I mean, say you found a skeleton laying around, or you were digging on your land and you find a skeleton. What are the possibilities? What could it be? Well, it could be a murder victim from 20 years ago. And if you've been living there for 20 years, maybe you'll be real interested in that. I didn't see any cars driving around then. Or maybe it's a pioneer who was buried on his way to Oregon. And or maybe it's a Native American burial that's been there for several hundred years or 1,000 years. Now, how do you tell? With bones, and only bones, well, the only way that you can tell is based on the features that you can observe on them and what's endogenous to them. The endogenous is radiocarbon.
Okay.
So you do radiocarbon and say, well, okay, this skeleton looks like it's 3,000 years old. It's got to be Native American. If you look at the features, they ought to comport with that. I mean, if you've got enough of the skeleton that you've got features that, in fact, vary between Euro, African, and Native American populations, you can say, well, yeah, I think these features are consistent with what I think the age of it is. Well, we do the same things with Neanderthals and other ancient groups of people. You've got a cave. It's in Europe. You found it. You dug down deep enough in it. You start finding these stone tool types that were only made before 40,000 years ago. And you find some bones in with it. Well, if it's just a piece of bone, if it's literally a tooth, the tooth might carry information on it about its morphology, about its shape. And Neanderthal teeth are a little bit different from ours in some respects. So the tooth might be enough to say, oh, yeah, it's a Neanderthal tooth. Or it might not be enough.
I don't know.
Well, nowadays, you've got that tooth or you've got that piece of bone. You can drill it.
I don't know.
Obviously, you can do radiocarbon dating if it's young enough, which is less than 45,000 years.
I don't know.
And if it's well preserved enough. It's got to have that organic carbon left in it. If it's got that, there's a good chance nowadays you can get DNA out of it. And so there are cases where we have bones that were found in sites that are far from Europe that have a Neanderthal mitochondrial sequence that we find in Europe, and it's very similar to Neanderthals. So we say, well, these are probably Neanderthals. This is their geographic range. There's a case that came out earlier this spring with a site in the Altai Mountains, sort of southern, almost. I wouldn't say eastern Siberia.
I don't know.
It's not Vladivostok, but it's it's sort of on the border of Afghanistan, China, that region. It was a pinky bone, the end of your finger, and pinky bones are not at all diagnostic. If you've got a pinky bone, you would never say, "Oh, yeah, it's a Neanderthal pinky bone." You would never say that. But they were able to get DNA out of this, and they showed that, in fact, it's quite divergent, the mitochondrial sequence, from the known Neanderthals and from any living population. This Neanderthal mitochondria looked like they diverged from us about 460,000 years ago.
Wow.
Which means that every Neanderthal that we know of and every human that we know of share at least one maternal ancestor who lived 460,000 years ago.
Wow.
This pinky bones DNA diverged from the rest of us 1 million years ago, which means that this bone didn't share a common ancestor with the rest of us maternally then, when the rest of us did, and the Neanderthals. It was much earlier in time.
That's amazing.
What that means, it's not entirely clear. This site's called Denisova, and it was in the news too, so somebody may have heard of it. What do we take from this? Well, it could represent another population of ancient humans that lived in this area that wasn't sharing a lot of genes with Neanderthals. Or maybe it represents a divergent group of Neanderthals that happen to have a different mitochondrial sequence. We know from woolly mammoth mitochondrial DNA that this sort of thing happened. There were different populations of woolly mammoths, and they were in some cases quite divergent from each other in mitochondrial DNA terms. And occasionally they replaced each other. So the stuff that we know now from paleogenomics that you would never have expected to learn. I mean, when I said we had a couple hundred good Neanderthal specimens, the end of that statement, of course, is, and today we have 20,000 genes.
Yeah.
and it's literally an increase of 2 orders of magnitude, the information that we have about them and their biology.
Yeah, the statistic I saw was 400 milligrams of bone to make, the 5 billion base pairs of DNA or something. I mean, it's amazing, just sort of. Yeah.
It's an aspirin tablet.
Yeah.
It literally is a tiny, tiny, tiny amount of bone. The technology that they've been able to, I mean, you can do this with living tissue, with a tiny amount, this genomics. It's the problems of the paleogenomics are that the DNA is broken up into tiny little bits. And so you have to be able to sequence every tiny bit. And that's really what the next generation sequencing has been able to do. It sequences the tiny bits. So it's a nice convergence of technology, which is being developed for entirely different purposes. I mean, that's the fast way to assemble genomes now, and this particular need.
Yeah, I thought actually that was sort of what I thought the connection sort of between these two stories, the synthetic genome and then the Neanderthal genome, sort of that they're both sort of these amazing, very complicated technologies about DNA. It was sequencing and synthesizing and sequencing again and then synthesizing more, and sort of and bringing together all this data. But I think the reason that become such big stories, even though they're more of these like sort of technological breakthroughs, is because they both sort of tell us a little bit more about what it means to be human and what it means to be alive. And I thought that was sort of really special about the stories, both stories.
Oh, I think so too. You think of the connections when Craig Venter is sailing around the ocean on his ship. I mean, the whole purpose of this, they say, is maybe he has other purposes. I don't know.
Getting a tan.
But the whole purpose of this is he's going to scoop up seawater and sequence every bit of DNA that's in it and discover what's there, right? This is exactly what we do with these bones. You drill into this bone, you take out powder, and you sequence every bit of DNA that's there. And most of the DNA is soil bacteria and fungal. So the computers sort that out. They say, okay, well, this is soil bacterium. Here's a fungal. You sort that out, and they focus on the human-like stuff. Yeah. And of course, there's all the problems of ensuring that you've eliminated contamination possibilities, so you don't have actual human DNA in it.
That sounds so hard. I can't even.
It's impossible. Yeah. It's completely
impossible. Considering how hard it is for me to keep, bacteria out of my experiments.
Well, see, here's the thing: is that you don't purify. You just sequence everything that's there. The only thing you have to make sure to do is to make sure that the woman in the Q-tip factory isn't, this is a forensic case in Germany where they had a serial killer. That kept showing up this DNA profile, and it was a woman, and it turned out to be that it was a woman who worked in the cotton swab factory. And the company was, these aren't we didn't ever say that these were good for sterile use,
Yeah. Wow.
But in terms of the paleogenomics, the thing that strikes me as the real commonality between these stories is that earlier this year or last year, late last year, I don't remember which, they reported on mammoth DNA sequences from the tundra.
I think I remember
Mammoth
that.
DNA is not especially new. I mean, we know that it's preserved in mammoth remains, and you can get DNA sequences out of them. There's a mammoth genome draft. But what was interesting to me was they just drilled holes in the ground and took samples in the tundra.
Oh, wow.
And there's mammoth DNA in it.
That makes sense,
And
I guess.
on the one hand, I mean, this is what they call metagenomics, right? You're going to sequence every piece of DNA and let the computers figure out what's there based on similarities to published genome drafts. And so that's how they can do this. They sequence every piece of DNA there. They find the mammoth because it's similar to what they know about mammoths and elephants. But the other thing is, just imagine how much mammoth flesh there has to be on the world to be able to drill a hole in a random place and find mammoth DNA. It really adds that dimension to the history of life that we've been recycling and recycling and recycling so much that random molecules that are still in the ground were parts of mammoth bodies.
That's amazing.
Yeah, it's incredible.
Yeah, I think the other connection that I think I see, sort of talking about genomes and sort of all this history of all these genomes, is also sort of show what more we need than just the genomes. Sort of like we have all this data and sort of and we can now synthesize a whole genome, but we still sort of see that we still need the cell, in the case of the synthetic genome, in order to boot it up and to have it become alive. And then I think even for the Neanderthal, like we get so much more information from understanding the genome and sort of seeing the genome, but we still don't know, what do those genes do? Why are we different? What are the differences? Like, where do we see them? So I think there's still, it's sort of, there's still so much more that we need to understand.
Oh, absolutely. In biology, there's this, or in genetics, there's this sort of division between what they call population genetics and what you'd call functional genetics.
Yeah.
And if, from a population genetic standpoint, somebody like me can say, all right, I've got this gene, and I know its sequence, and I know about the transmission properties of this gene. I know how fast it mutates, more or less. And so I can say things about the population from this gene. I can even say, in many cases, whether the gene has been important to fitness. I can say, has it been selected? Has it changed faster than it should have? Or is there a commonality among people that shouldn't be there unless there was some important function? That you can do with just a gene sequence. But that really is a very limited set of questions. And when we say that, yes, we now can confirm that Neanderthals contributed some fraction to the human ancestry, that tells us a lot about our species. It tells us what its boundaries are. They're bigger than we thought they were. It encompasses more of the ancient human archaeological fossil record than we thought. But it doesn't tell us almost anything at all about what these ancient people were like. And there are these hints that we've got in the last few years about the function of Neanderthals, as an example. They recovered a gene called MC1R, which is it's the melanocortin-1 receptor. What it does, among other things, is it makes people red-headed who live in Europe. Because if this receptor doesn't function as efficiently, it prevents the normal process of making eumelanin. It prevents it from happening to some extent. And so the precursor molecule, pheomelanin, which is red, sticks around in these melanosomes. Well, we know from the Neanderthal sequence of this gene that it didn't work as well as the normal version. It has a mutation that would have interrupted the receptivity to the hormone. And so we can infer that functionally, Neanderthals should have had red pigmentation in the same way that human redheads do.
Was that study? So was that just known from the sequence, or were we people sort of like doing experiments on mutated genes in cells and looking at pigments?
So what they did was they took the sequence and they confirmed. I mean, with ancient DNA, which is damaged, you have to confirm that the same mutation really existed. So you got to find it in multiple individuals, and they did that. So they've got the sequence, and then they say, okay, this is different from the wild-type human sequence. It's not one of the red-headed alleles that we now have. It's a different mutation than that. So what can we do? Well, let's look at the part of the gene this is in. And they said, okay, it's in the part of the gene that's responsible for receptivity, and based on the amino acid substitution that it would have caused, we can do a reconstruction of the protein structure. We can show that this wouldn't have fit as well. So they've got this sort of functional prediction of it. Now, of course, the next thing that you do is, which they haven't done, is take this gene and put it in a mouse and see that it actually has that effect in vivo.
Would the mice be red-headed? Is that the red-headed mice?
Exactly.
Okay.
You do this with, think of what you have to do functional genetics.
Yeah.
To say, we know from the sequence what the difference is to the enzyme or to the molecule. And we know from that difference that there will be this phenotypic effect. Wow, we're really at the very beginnings of being able to do this with anything. I mean, it's really a nightmare.
It's a lot of work.
It's a lot of work, yeah.
I think actually that's sort of something that brings back to sort of they're talking about eukaryotic synthetic biology. There's more people who study yeast genomics than there are genes in yeast, but there are still uncharacterized yeast genes, and sort of like this is very common because there's more people that study Neanderthals than there are Neanderthal fossils. Yeah, but you sort of get to the point where the ones that are uncharacterized are the ones that don't have an obvious function when you delete them, and don't sort of look like other genes that have known functions. And so, like the kinds of experiments that you need to do to sort of fully characterize an organism and at sort of a reductionist level based on their genes is incredibly time consuming. That's each one of those genes is several PhD theses, and so.
Oh, absolutely. Yeah. I mean, in humans, the genes where we know more or less what they do are things that are visually obvious and that are part of well-described molecular pathways. So things like pigmentation. I mean, why is it we know so much about pigmentation? Because there are so many white mice in the world. And many of them are white for different reasons. And so we know that when we break certain things, it has effects on pigmentation. All kinds of mammals have pigmentation variants, and so we can look at them in an evolutionary context. And in humans, we have a lot of them that are under recent pretty strong selection in some populations, so they are visually obvious to us. And we can characterize, we can run gene chips on people, find the genotypes that are associated with lighter color. We can do all that stuff. I have a graduate student now who's focusing on diet. And he's real interested in the differences of diet adaptations that have emerged in Neanderthals versus modern humans, and also in different human populations that have a long history of agriculture. And
is that stuff like the lactose
genes,
intolerance and stuff?
So lactose is the
classic.
Easy example. We've got this gene, this enzyme that helps us break down lactose. And if you don't have a persistence of this through your life, then as an adult you become lactose intolerant, we say. And that's the normal, that's the wild type. But most things are not nearly that simple. They're not cases where there's one enzyme where it's broken. Most cases are parts of really big interacting networks, and we don't understand them yet. And so this is a tremendous problem. And hopefully, from my students' point of view and from mine, hopefully we figure out what has changed, and that gives us a clue to what might actually be important. And starting from that end, you figure out, okay, we know that this has changed rapidly, so it's important. So it must do something, and the logical, plausible things for it to have done are this. And then you move to the next step. Okay, test to see if it does that. It's a long process.
Yeah, that seems so. I mean, that's so interesting because so much of diet too, though, is sort of caught up in culture, right? So you would sort of do you think that you'd be able to find sort of trace cultural shifts too, and as populations diverge and see, start eating different things, also depending on their...
I expect that we will. I mean, one of the big polymorphic gene systems in humans is olfactory receptors.
Oh, that's really
there are
cool.
some things that some of us smell that others of us don't smell. And probably the most famous of these is the asparagus pea gene, right? Because some people, when you eat asparagus, there's a molecule that goes in your urine, and it has a very obvious smell.
And some people can't smell
to
it?
some people. Oh, yeah, and some people can't smell it.
I didn't know that. I thought that was a universal.
But there are any number of these genes where some people smell it and other people don't. And that's got to have an influence.
Is that because there are different receptors?
Your food
or
preferences.
or the receptors are always there, but there's mutations in the... Or they're just like, is there a different set of receptors?
In some cases, they're pseudogenes.
Okay.
They're genes that in some people are completely broken. In other cases, that may be some sort of polymorphism. But
I don't know.
Polymorphism in every case. But in some cases, it may be a functional polymorphism where it's not as salient.
I don't know.
But yeah, that's got to have an influence on tastes.
Yeah.
And to the extent that they're different in different populations, you've got to figure that this interacts with culture. So that's the kind of thing that, as anthropologists, we really want to be able to uncover and explain the history of.
That's really interesting.
Okay, well, my clock is showing that we've come to an hour, and so we've got to wrap this up. Any final words?
I don't know. I think we sort of ended up talking about everything that I had been thinking about. So, what are your final words?
I think we've done an excellent job. I mean, obviously, there's a lot of stuff on both of these topics, the synthetic biology and Neanderthals, that we could explore for hours and hours. And obviously, if somebody is interested in those topics, they should check out your blog and my blog, because we're writing about them. And hopefully we'll get to have a conversation like this again.
Yeah, this is great. I never thought there would be so many connections between the... I never thought there would be so many connections between the...
Well, that's the way it goes.
Well, thank you.
Thank you.
I never thought there would be so many connections between the two topics. I don't know. I never thought there would be so many connections between the two topics. Yeah, this is great. I never thought there would be so many connections.